And actually, it would help if I shared the companion. Right. So yeah, links in a link to a form. If people have the inclination, it's just so options, and we can hopefully improve it in the future. At the end of BGA feedback form, in case there's anything you'd like to see done better in the future, to have a laugh. And that's actually the wrong one. It's that one, EBP 1. Right, and there will be a bit of noise behind me. I'm in the conference center. All kinds of fun.
Two minutes later.
Yeah. Okay. I'll let you go for it because it's your workshop. Normally, I've been doing like an intro to each of these, but no, this is very much more your realm.
Okay,
so, please go for it.
Oh, yeah, it's everyone in the workshop. And maybe I could share the screen. I think everyone already see the screen. Emma, can you see what I share?
Yep.
Yeah, I'm listening to another Biodiversity Genomics Academy forecast in the background.
Maybe I think. Okay, so yeah, happy that everyone is the second workshop of the first one is held 2 years ago. So there is, I think, there is much informatics in the genomics research. So I quickly will talk about the agenda of these 2 days. There is 4 hours. Today we have a section 1 and best practice and the challenges. And in this section, will including the best practice of the I5K standard bio headers about the data accessibility and all the other design about the bio headers. There are some, maybe they have some tools and the insights from the NCBI team. We'll make some discussion about medical data repository concepts and also the management principles. And this section 2 is about the genomic. Okay, which is large screen. Yeah, the section 2 is about the genomics informatic tools and database, because our the EBP, the IT and the informatics subcommittee will focus much on the informatics things, and just like the trace or in some other platforms. So in this section will be our main part, some key resources or platforms or systems. The first one is maybe someone knows of the the trees, the goat is a centralized search engines, the information about the genomes, and also will we will make some introduction on the new platforms developed by BGI teams, and also the Galaxy platforms. Many people are quite familiar with the Galaxy, and hope that we can from this section everyone can get more about all is a roadmap or any new design in this system. About the genomics informatic tools and databases, and we will have made some discussion of writing the pipelines, the tree lines, example, and Emilio introduction of the BGP portal, and Terence so make NCBI toolkit and to how to use. And the fourth one will still make a couple of some biodiversity portals of the EBI, and also genome technical things, how to analyze the genomics data. We right now have some concepts of the develop a system we call the BioGenome. This part. So from today, and today we have section 1 and section 2. Part about the EBP and our standards. Yeah, the code homepage, and it's quite useful tools projects, and maybe other projects is also work well. DC because there is a EBP umbrella projects. The useful informations, what kinds of species already sequenced, and what you need to do, or what kinds of species you need to sequence in the future, and also the progress of all the projects in EBP, and also the EBP IT and informatics standards. And right now we already have visions, and the latest vision updates last year. And I hope that after this workshop, we can compile all these reports and all these topics, and make them as the best practice to these EBP IT engineers. And because you may know that IT and informatics, it's right with the sample collections or some sample management system or some sequencing tech or some data analysis pipelines or some or even some other kinds of details you do in your research or genomics research. It's a little far away from them. Sometimes not much teams they focus on this part. So I hope that after this workshop, all these we can move these genomics standards in the EBP forward. So I also put because I like sometimes because I photograph every day. So there is Stephen Shower. He's an American photographer called Uncommon Places. And this picture is one of his favorite pictures. And there is some new concept from his photograph, but I don't like there. He has a words that sentence does not. So I think that it's the same set to our genomic that from this workshop everyone can get what you can get. Okay, so we just start, and now I just stop the sharing. Professor Mark from the University of on the genetic and regulatory that maybe some and his teams also focus on the by had developed maintains a database called a red is internationally right now, and also it's a community that ensure that all of the insights some in the future can perform or can be used across different inside scientists. Yeah. So Professor Mark, maybe you can share your presentation. Thank you.
See if this loads up. Okay, how's that? Oh, thank you, and good morning or afternoon or evening, I guess, depending on where everybody is joining in from. It's morning for us. Yeah, I'm going to speak today as a representative of the I5K Standards Working Group. And what I'd really like to do is deliver sort of an interim report on what we've been past year. So this is very much a work in progress, and we're very much interested in feedback from other members of the group time at the end, or people can contact us, of course. Here's just the members of our working group. Like I say, I'm representing them. This is a very much a group effort. And I thought I'd just start with a brief introduction to the I5K group. The I5 is a community that was formed about in 2011, 2012 with the idea of facilitating of initially 5,000 different arthropods, primarily insects. And come to that goal. We now have over 6,000 arthropods that have been sequenced, including over 1,500 chromosomes. 2 years ago, as we started reaching that goal, we started talking to our members, and this we are well distributed around the world. We've recently done some reach out and participating in I5K coming from there as well. But we've reached out to our members about 2 years ago. Now that we're sort of nearing this initial goal of this many arthropod sequences, what should the I5 this vision statement and mission statement? The idea is that the I5 is clear that these genomes are available to all researchers who need to use them. The methods and the standards that are needed to work with these data are available to the whole community. That we would promote the best practices for genome projects, support the generation of genomic and post-genomic data, as well as present recommendations to ensure that we have the support that we need to continue to pursue these goals. Different steering committees, and today I'll really be talking to you about the standards work. Come up with guidance for genome projects and to publicize best practice, new standards as may be needed. This came who clearly requested this type of guidance to work on their we conducted about 2 years ago. And you can see, I won't go through the whole list here. Really, we're looking for, could we make recommendations on specific techniques? What standards are out there? What are the best ways to deposit the data? Where should data be deposited, and sort of what formats should be used for that? So there was a lot of need from the community for guidance on these issues, and that's why we formed this working group to discuss standards. Initially, we decided that the place to start would be by just conducting a sort of gap analysis to say, well, what standards are out there? What standards are missing? What practices are in use? What practices, need to be better publicized and incentivized amongst our community overall, but in particular as that obviously pertains to genomics, or are there special considerations for arthropods that we need to have there? And to do that, gap and follow the genome workflow that was laid out by the Earth BioGenome Project. We broke it down into all these different areas that were laid out by that group: funding and budgeting, sourcing and vouchering, sequencing, prediction, databasing, various downstream analyses, and just sort of brought a group together to start looking at what gaps there would be. So we met a little over a year ago, our first group, and we had various domain experts in these areas who would have expertise to lend us. And the idea specifically what we need for arthropod genome projects, but also what may be needed in general there. So what are the existing requires modification for our group, and what standards or practices are really lacking for in particular. So the first thing that we realized is and a lot of this is thanks to all the excellent work that the EBP has done, and that are through everything, but sort of you can see on the right of these slides as I go through them that there are a lot of existing starting point for this. And what we really did not want to do was already there. So we had very good starting points really for all of our areas, for sequence, databasing. Those all existed from the EBP. From this, that there's some gaps that we should be considering. One would be what we decided to call where the standard or metric that we need really doesn't exist yet. Right, so some EBP that we could rely on, or those standards didn't community there. And so these would be cases where new standards need to be developed. Okay. The other type of gap we realized is what we decided to where those standards do exist. So the processes and metrics that we need community. And so these are places where we don't necessarily need to develop a new standard, but where our community needs to be educated and or incentivized in order to adopt these standards and make use of these standards. And in fact, we found this to be much more prevalent than the lack of standards altogether, again, because so much work has already been done by EBP and other groups. What we also realized, although we were really focused on our community, a lot of the gaps we identified really weren't arthropod specific, but were sort of generic genome project issues and places where overall there weren't good standards or those standards weren't being widely adopted, not just within our community, but in general. And so with sort of a narrower focus, we think that it's sort of broadened out as we've done this, and we think that the information is going to be of broad use because it's going to affect much more than just the arthropod community. Through what some of these gaps that we identified were in our visioning process. Again, this is just an interim report. I'm going to go into a lot of detail on this. This is just to give an idea of what we have. And we sort of out whether these are practice gaps or gaps in the standards themselves or some combination of both where there need to be new standards, but and so we started with the budgeting and funding category. And of course, there aren't necessarily direct standards. So to recognize that in practice, you know, some projects start out without really having a good sense of and what needs to be there. And we thought there's room for guidance there for people who want to embark upon a genomics project. And of course, I'm talking about smaller individual groups who might have a organism of interest that they want to do. And what we recognized is weren't really in place and carefully accounted for in the beginning. A lot of these projects would peter out, never really, but not really a good genome. And so this is an area where we aim for data management plans. There's some research that shows that when a good data management plan, a lot of time data end up not being easily accessible or widely accessible. And sort of standard template that people could use, but also education about how to use that and do it, understand where to publish the data and what sort of formats to use. Standards and practices that we recognized. One issue with round is what should replace things like the N50 measure. Long read sequences where we expect the context to be really long and for there to be relative. The feeling was that the community in general hasn't really coalesced upon good alternatives. Needs to be sort of with everybody in terms of standards that can use Kamer-based methods and things like that to get good measures. But we find that often these aren't being used by people in their projects. And so that's a place where the practice needs to be improved. The same is true for assembly correctness. Curation, contamination, there are good standards and there are good methods to take care of contaminating sequences and sequences from cobionts, but not everyone is aware of these, not everyone uses these, and you find these sequences creeping into assemblies and all. And so that's another place where I think the technology exists, but we need to make sure that it's being widely adopted by all users in the community. In terms of analysis, is something that's very important, obviously. There are good workflow managers, and there are a lot of things being done in that area. That in our community, at least in the arthropod community, it still needs to see wider adoption and using and reproducible workflows that are out there that people can then make use of and reanalyze data and analyses that needs to be improved upon. Annotation, again, gaps in both practices I noticed, there are good methods and standards out there for dealing with repeat annotation and gene these adopted by all members of our community. Something near and dear to me is regulatory region annotation. Not have good standards for doing that. It's not clear what are the best ways to do regular. It's also something that's often overlooked, and most genome annotations that get published really don't include much in terms of the focus on the coding genome. Quality assessment place, I think there are need for additional standards and additional work. So BUSCO and tools like that are often at least weighted towards dominant model organisms such as Drosophila, and therefore, as we said, members of the phylum, that those tools of the specific genes that are there, and so we think there's room to find that need, and that goes for quality assessment too, as well as nomenclature. Again, Drosophila, that doesn't bother me particularly. I'm a longtime Drosophilist, but names and functions and identities of genes and gene products of that referencing and so forth. That is a problem. So transferring annotations and knowing which annotations to from and how to, there is a standard, obviously, GFF has a standard, but it's obvious to do your annotations in a GFF file. And we find that those my own breaks a lot of software pipelines because things are need better standards, we think, and we need to publicize those and work for them to be more widely adopted. Databasing is a big problem. So there are a lot of databases out there, but some of them can be hard to find. Maintain smaller community databases is an ongoing issue, and what happens is things then disappear or become less fun, and they're not always permanently archived in ways that lets someone access data once they having difficulties or for whatever reason. We need better standards for interoperability among these different frameworks and different identifiers that get used. If that would be more standardized, then we could cross databases better. So there's room for standards there, and also practices. So there are certain standards. It would be useful if those were more widely adopted. There are some database hubs that can be used. The Library has the I5K workspace where people can deposit their insect genome projects, for instance. Takes advantage of some of these pre-existing formats and pre-existing software that's out there. This is more of the same, really, both in practice and with standards for that, would be very helpful in terms not just and gene functions as well. That's and sort of where some of the key gaps are, and there's other stuff in there. I'm not. So in the short term, we're taking the following steps. Outlined where there are standards gaps, where there are practice gaps. And we hope that should provide some talking points and some reference points. So in undertaking an analysis of genome reports and real data gauging the incorporation of those standards that are out there. So places where we'd like to gather some data to really see what the extent of sort of an interim step, working on a manuscript describing submitting journals that publish these type of supports to find out what sort of data, and we'd like to compile that along with our recommendations of additional and then in general sort of we have at least we're interested in developing educational address some of those practice gaps a lot of standards that are out there or don't have the proper incentives to make use of those standards that are out there. And so I think by educating the community and with some of the publishers that we can make a dent in some of that. The other long-term item that we need to do, obviously, is form some to develop those standards that are lacking that we've identified that are needed. And we hope to begin doing that in the future when we analyze our gap analysis. And for that, we will almost certainly be reaching out to other members of the community, not just arthropod people, of developing good and proper standards in places where they're currently lacking. Late, so I'm going to try to wrap up a little bit early so we can keep on schedule here. I just want to thank the group as well as all the participants in our initial gap analysis workshop.
Workshop? Maybe job, right? Yeah.
I lost
okay.
the Zoom there. Let's try that again. I don't know what happened. I was just trying to thank everyone who participated here. And I apologize if I left anybody off of this process. But this is being able to do this work. And I thank as well all the members of the I5K consortium lent their expertise and has also lent their questions. And with that, I'd be happy to answer questions or take a little discussion in pushing this forward and knowing what people think about some of these gaps or recommend people who are interested in working with us, please contact me later on. And we're
Thank you. Thank you, Professor Mark. So, okay, anyone wants to raise your hands? I found that Mark, your standards cover and even in the future application. So our teams or maybe some other teams also focus on some image.
Yet we are aware of it. We also know, as you say, that there's an important aspect as well. I didn't talk about special yet, but working on. So.
So any questions? Maybe you can contact Mark later. So I will send the mail.
Great, thank you.
To everyone. Yeah, thank you, Professor Mark. So yeah, speakers. I thought Davey and Adams were online. So both of you made a.
It's me today.
Okay, yeah.
I get confusing, I think.
Thank you. Questions. And Dr. Davey and Dr. Adams is from the United States Department of Agriculture. Also read the papers about the failed biohazards of reference genomes. And I found it because right now debate by more and more people and more and more teams. Yeah, and they have SARS and hope that everyone, attend. So okay, Davey?
Absolutely.
Please.
Yeah, let me
Yeah.
share my whole thing in chat, which will be useful for the share slideshow. I think I'll start off with just a bit of an introduction of Adam and myself. So I for the United States Department of Agriculture Arthropod-Borne Animal Disease Research Unit in the gene regulation in agriculturally relevant events, which have their own problems. And I would love to talk about which is data standards and perhaps filling. Adam is a software architect for technical science of genome resources, including Wormbase, and of course, Betters project. I've posted our relevant-ish manuscript for about this same problem in other spaces. There's a problem with copying data from a central data repository institute's high performance computing center or confusion in the reference genome. One data repository directly to where it's analyzed, but from one data repository to a high performance computing center to somebody's desktop and maybe around a lab. This recursive sharing can introduce various changes, sometimes due to copy errors. Large genome or large files in generally can have packet loss, which will reduce the file size. This is one of the principal reasons that people started thinking about checksums on reference genome assemblies. But we've also noticed that when these files are getting shared around, sometimes what can happen is that there will be small edits to the file, and so that the chain of what we'd call data integrity is lost. So thinking about this, we wanted to create a standard which could, and when I say standard, I'm using that in the loosest of terms. We're talking about a schema, associated serializations. And I would more often than call it a serialization and not a standard. There's a big difference. Only the schema is a standard. Everything else is a serialization. But what we wanted to do is create a standard that could minimally invasiveness, a light touch, preserve data provenance from the system. And we wanted to do this in such a way that at any point within this chain, we could. So, and you could think about this in terms of perhaps, you have NCBI being sort of linked to by one linked to some JBrowse instance somewhere, or an Apollo, or the Apollo instance continue system. So there's two solutions to sort of keep data. One, I already referenced it with checksums. So a checksum is just a hash, lossy hash of the data itself. And so you can button, I check that my file is validated. Another way to the provenance. And a collection of feasibly what the file that you're looking at is. Where it came from. To do this, several serializations, we use schema as separate tools, but then imported as Python libraries into other tools that can utilize the validation and creation that we've written. So you can essentially import it into whatever. A key component of this is the algorithm checksum. As I alluded to, it's a string representation of a document, each document a unique string, and it can tell you whether or not it's complete. So if you have data packet loss, it would tell you that, and then that created a write error. Actually, sometimes what I've seen is that with large files, not to scare anyone in the chat, with large files, if you're writing to a very large file, there's a data backup problem, and you'll actually miss a component. One place where I saw this is I was using an API, won't say whose, and I was hitting different ways in four different analyses. And so when I expected this of returns from each of these APIs. Of course, over 1 million hits, right? And 1 million times 4, so 4 million times, those files did not match up. So I have to go back, figure out what got missed, and then reanalysis. Now, the easier way to do that is, through validation checking in your API calls. You should be doing that. But again, not paying attention. And a checksum can also tell you whether or not that file is the same as another file. And so if you've got two genomes, G17, one is a GH, I'm not a human researcher, but they're, the same human genome is this, but they have two different file names with two different, sort of provenances, then a checksum could tell you whether or not in any document recording. So FHR also works on recording provenance, and so it'll record things recorded in that the author of the data, but a different team might be annotating it within an FHR serialization, and so we split those off. Our hope here FHR instance somewhere would go a long way to recreating that genome assembly from scratch. It's serialized in different formats, YAML, JSON, and microdata. As inclusion in the FASTA itself, and we do some interesting to validate and convert between formats. And we again, and I can't stress this enough. We work on this idea that you have to sort of be strategic about where you want to spend your time, and and so we strive to lower that action potential as much as possible. Just here, use this tool. It gets you most of the way. Then all you really need to know is the information. Suddenly, you've got a much more data integrity, a much more much higher data integrity. So the main way that we hope that FHR is implemented is through the FASTA format. So FASTA, created in the 1980s, it's been around for a while and hasn't changed much. Greater than sign, which talks about the header of a contig, a sequence for contigs and scaffolds, and we use it in different ways. And what is that in the original specification of FASTA, there actually was a comment line, and the comment semicolon. Of course, not nobody, but very few people use line today, but some tools, I'm thinking of like the R sequencer library, which will ignore the semicolon. And so all of the metadata in these comment lines to at least reduce the hurt to FASTA files, making it slightly easier for an environment where you'd have these in multiple. But we also, in order to control for that, have written a tool that file if it's absolutely necessary. So it should be in the FASTA. We want it to be, but might be kind of a tough call, and so, or a tough thing to do, and so we've written to lowering the action potential of adoption. Other microdata, which is basically HTML tags, but a certain way to use them, or can use them. That comes into play in our sort of next steps and future about that. So this minimal mentality. So we want so it continues to exist with the FASTA in perpetuity, but we provide stations to share and board. FHR records necessary metadata. That could otherwise be assisted. It also records the checksum within the file. So we have that we wrote a whole tool to control for that. And the file itself. Some of our synonyms for the genome names, we recognize that sometimes require identifiers.org taxonomy, the metadata author and assembly author, which I talked about, they created physical samples, which is actually turned into a new field called voucher. All of your reference genomes should be vouchered somewhere physically. And if it's not physically a voucher, it should be from, the location that the assembly was created. That's the sequencing center, instruments, assembly software. Come to think of it, everything after instrument is not a required field, but there to increase the amount of provenance in the file. We go through a lengthy debate every time somebody asks for a new field, and we do discussions. That field would really increase the usefulness of the file versus increase the sort of clutter of the header. This is not intended to be the full description that you might have somewhere, to be the minimal requirements. Again, can't stress that enough. So this is what an example had the semicolon, and then we use a secondary character, and this we use the tilde. This denotes just regular old comments from the header. And so the header can be anywhere within the file. It can also be the validation software will just strip all of the lines with the tilde, and we there's a whole story about why a tilde, but you'll have the data, which is then imported into the Python library. It could be used in further analysis if so wanted. Find this under the auspices of FAIR principles. Now, this is a scheme and standardizations or serializations, but we wanted to itself cannot be fair because only data can be fair. But we wanted to ensure the standard and the serializations were increasing for the data ability to reach trust model I get into. So we go through and we look at findability, accessibility, interoperability, and reuse. A fair bioheader. We also do that with trust, which is transparency technology. So have started embedding the ability to use fair headers into other software files. You can it will strip the FHR embedded server concept site where you can press this and it returns the fair bioheader information. You can up or and sidecar. We're going to be many all of the provenance are pushes to standard with in the Sanger Institute, which will tag that table with all of the provenance information, the genome notes, and then a FHR header will be able to return from that genome notes table. With that, special thanks to all of the co-authors and all of many fruitful discussions with. I won't go through the entire list, but needless to say, very fruitful discussions came out of all of this. With that, I'll, in the interest of time, I'll take any questions or any discussion.
Hello, David. Did you hear? Oh, yeah. Thanks for your talk. My name is Raymond. I was working on the whole and the company. And then one thing I'm not quite sure. 100%. I'm not sure whether I understood correctly because in one slide you mentioned that if I lost my genome assembly, for example, I instant, but if I have the FHR instant, I can use it to with genome assembly because I have put the pipeline or how to produce my genome assembly input. I'm not quite sure whether I understand this correctly. If I'm wrong, you can correct me.
Yeah, I alluded to being able to regenerate assembly from an FHR instance. The GATCs, but having the ability. So this is not computationally. This is the worst this happens in several areas that have reduced the structure to. This is obviously never going to, hopefully never going to happen, but the file, I know what at least was done for that file. I know who created. I know, what tools were used to create it, and so back to extractions again. Go to the sequencing core. The assembly tools software, I would be able to do that, or I would know the information of all of the stuff involved and be able to run the experiment again, or run the assembly that we're able to regenerate the contigs and the scaffolds. Some recent catastrophic in the worst way. For which to look at used to, do we really need this field if we know?
Yeah. Thank you. Thanks, David.
Okay, thank you, David. And I think we need to move to the next topic. And yeah, thank you, Kim, for waiting so long. And Dr. Kim is acting director. We're happy to invite her to join this workshop. And the acting director of the SBI, and used to be a chief. I get the info from the web page that used to be the chief of the information engineering branches at NCBI. And today we will share some about medical data repository concept. But maybe everyone think that this from because we focus on some biodiversity. But I think even in China, I'm not sure that every country research or some animal research, we finally transfer the data. We find some useful genes can be used in medical research or biomedical medicines things. So I'm happy that Dr. Kim can share his ideas about this part. So go on. Yeah.
Thank you.
Thank you.
See the right monitor?
Yeah.
Sure.
Yeah.
Okay.
Okay.
Thank you very much. Good day, everybody. So introduce me. I'm Kim Pruitt, Acting Director of the National Center for Biotechnology Information, otherwise known as of the National Library of Medicine at the National Institutes of Health. And so the publication, I stuck it in the chat so you can read it for yourself. And you're going to be going over some concepts, some principles and properties, management best practices, community resources that are really about repository management. But not a lot of people talk about best practices around managing data repositories today. The definition, and this is the work of the NIH staff that different ICs at NIH. And so a data repository is access of data from researchers or public health groups or others and provides services and redistribute the data for reuse. In contrast, NIH defines a system for data sets that are managed by data repositories. Focus on extracting meaningful knowledge from these other information sources. Is something that we use to refer much more broadly to a specific purpose. And so it's an umbrella term over knowledge base and source. Incorporate and manage collections of discrete related data objects. These can be classified into different types, which are shown here. Domain-specific repositories accumulate a specific kind of data. So it's really as it's data, sequence data, but the data type is specific in those kinds of repositories. Generalist repositories do not have specific types of data. They are organized around a very broad type of data, anything that you want to put into a generalist repository. We also recognize that there can be project specific, and so like a data, like there could be I5K as an example, could have project specific data management and repositories that are organized around the needs of the insect community. And then there are also institutional repositories. And so, for example, an academic organization, a university, they may set up a universal an institutional repository to manage the research outputs of the scientists who work at that university. Repositories can be further described by these different kinds of characteristics. What community do they serve? With the community, what level of curation do they do? How do they preserve data? What other what type of data do they manage? And access for repositories can be open, which is anybody without registration needs. It could be something that requires registration. It could be controlled, as we see sensitive human sort of genomic data. It could be behind a paywall. Repository that a model where the community has to help support them by paying some, but sometimes it's required, or it could be proprietary.
Sorry, Kim.
Yeah.
Sorry, Kim.
Is there a problem?
1 minute.
1 minute.
Yeah.
Okay. The paper talks about the intersection of these items in the 1 minute. I think I'm just going to end here. I'm going to
maybe you can share the screen again. Yeah, maybe. Yeah.
Oh, I thought it was. Yeah. You see, yes. Okay. Yeah, so if you want me to end in 1 minute, I'd like to end here. Life cycle for biomedical data repositories. This is something that is, and we also describe how these things intersect, fair repository, how metrics are core to being able to assess the health and how it is meeting the trust and fair principles. And I will probably you later on about any of the elements because I think I'm out of time, right, Vincent?
I'm sorry.
Yes,
keep going. Yeah.
Use your time. Yes, so we are sending the link to the people in the second session, so they are joining now to actually see the end of the talk. So it's all good.
Yes.
Sorry.
Sorry, I just there is 1 minute we cannot see the screen. Yeah.
Oh, okay. Sorry. So the trust principles have been described in a publication by Dr. Lin et al. And these are principles for management of data repositories. And so, repositories need to be transparent. They need to have good publication describing their services and the data holdings that they have. Repositories hold a responsibility to the community for the integrity of the data that they're holding. Repositories must maintain a focus on their user base. It's not, they need to make sure that data management norms and expectations of these communities are being met. The data dissemination is serving the needs of the communities that they're supposed to be serving. Repositories are efficient and effective in their data management and IT practices in order to be optimized chance of sustaining services and preserving data for the long term. And of course, repository technology, they need to be able to continue to say that the services they're providing are secure. So following the publication of the trust, the U.S. office, the White House office of OSCP, Office of Science, published the desirable characteristics for data repositories, and the link is here on the bottom of the slide. Also published guidance around desirable characteristics for data repositories. They can be categorized into sort of three kind of buckets here: organizational, and it's free and easy access. There's the maintenance of submitter clear documentation about using the data, the retention policy, and that the carrying out risk management practices around data management that are persistent, collecting and maintaining, providing QA, QC, curation services on the data to it. All of these are to support downstream, the fairness of the data. By elements in order to help support the fairness of the data. So here, I did want to give you a little bit of an introduction to the cycle of the data and the trustworthiness of the data repositories. Life cycles have been proposed and described by other groups, and I'm not going to talk about that here today. But we found very few descriptions of biomedical data repository life cycles. And so we adapted existing, the standard product life cycle description for the health sciences. And that's what's shown here on this screen. And so the introductory phase involves the initial development and establishment of a repository, which is perhaps being generated to meet new scientific needs. The first introduction may really be a minimum viable product with only a partial set of features. User feedback is gathered during this phase, user testing is solicited, and the feedback from those users informs on the growth phase, where additional features are added so that it's moving viable product into a operational product during that growth phase. The addition of features is based on input and feedback from the community about what their needs are in a product. And during this phase, the repository sees an increase in usage and community adoption. And during along a time scale, but the length of time in each phase is going to be variable depending particular that's being developed and introduced. Repositories during the growth of the data holdings and improving the user experience through enhanced accessibility and other kind of. At some point, the growth phase sort of, tailors out into a mature phase. This data repository has achieved significant use to be optimized, but the rate of change and the rate of user slope has decreased into a flatter part of the curve. Lasts or decades, or so the length of time that something may be, or variable even. And repositories, different kinds of. I'll give you an example. Sort of a sequence. Technology was no longer well. This technology. And they're going to use, I don't want to come here anymore. I will user interface and so that technology was not to keep up with technology changes. Result of seeing that starts to go down. Funding or a decrease of funding. With the PI leaves time phase, complex phase. And the decision point is really assess that when the repository managers or the organization and institution needs to assess that repository's future trajectory. Does it still meet a scientific need? Is there a way to stabilize the funding? Do we have stability in staffing to manage that repository? And they need to actually make a decision whether or not to sunset that repository or to reinvest in it, such as to modernize the technology, modernize the user interface, change things to better meet the current user needs. And so this repository lifecycle model, I think, works very well for NIH repositories, and I think it probably will work well for other biomedical repositories. And of course, the last important piece of this is metrics. And so metrics are key. And so repository managers are utilizing metrics in they're utilizing many different kinds of metrics for several different purposes. And so they're understanding usage trends, they're understanding user behavior. And so is usage going up or is it going down? Are people staying on the pages for a long time? Are they doing multiple queries? How many users are going to the page? What are they clicking on the page? And so this user behavior category is like one of the core categories of metrics, right? How many times are they downloading the information? Where are they coming from? How global is your user base? This also, metrics are also used to assess or to try to assess, the level of contribution and impact of the data that's being held. And so if you're looking at something, you might be asking, well, what are the number of projects or studies that repository is receiving? And what are the number of cases or subjects in those projects or studies that the repository is managing data for? And everybody also tries to do some level of bibliometrics, which is difficult. It's the best we have right now for assessing scientific impact of repository, is by seeing how many citations that repository gets in papers. But often repositories are sort of taken for granted and they aren't cited in publications, and so it's not a satisfactory measure for scientific impact of that repository. And this is an area where much more research is needed. There are gaps here in being able to really understand the impact that repositories make and the holdings of the data they're holding. What impact does that repository have on society, on public health, on the advancement of knowledge, right? And so more metrics are needed in that area. Metrics are also used to understand repository operations. We're looking at what are our storage costs? What's it cost per data set? How fast are we processing submissions? How much does it cost per download? What can we do to make this more efficient? And making that more efficient makes the repository more sustainable. And there are numerous community resources in this space, and just a few of them are mentioned here. And these communities are working on elaborating best practices, elaborating on standards. And so standards around how to manage data, or standards around data formats, right? That can help make repositories more sustainable and more efficient. And standards about how do you know if your repository, is meeting the desirable characteristics, is meeting the trust principles. I'll highlight Core Trust Seal. It's a certification. You can go to Core Trust Seal and actually have your repository certified as a way of publicly communicating, that your repository is meeting the trust principles. Make Data Count is an interesting community. They are working on elaborating on different kinds of metrics around assessing data and assessing impact. And Research Data Alliance has also been very active in that space as a community formed organization. And I'm almost finished. And so, here's my my bottom line, right? Repositories, I feel, repositories in their operations, their trustworthy operations, are essential for scientific research. They're essential for both preserving the knowledge that comes out of scientific research, redistributing that knowledge so that others can reuse it, so that we can build upon, we're constantly building upon what we already know by new research endeavors. The repository landscape has been, is, and will be very diverse and continually evolving. Best operational practices are now emerging for trustworthiness, lifecycle management, and assessment metrics. And community efforts on all of these things, the information that's relevant to repository management, continues to mature and evolve. And so I think this is a very active space. I'm really excited to see some of these some of these principles and best practices starting to come out and be adopted by other groups. And I'm going to end there. And I'm happy to talk to anybody further about this later on. I know we're over time and we should move on to the next talk.
Okay, thank you. I really learned a lot from your presentation, because, National Gene Bank, I also have a teams focus on data repository, but never source. I think we never deep thinking about the data repository, the life cycle. There is a model of life cycle, because I think maybe I think in the future you can make another presentation EBP because I think most of the projects in EBP, they have a repository, they have database, but they have no framework or concept, they how to manage their database. Because right now in the world, there is many bioinformatics database, but 3 years or 5 years, they close their database. Yeah. Great. Thank you, Kim. Yeah.
You're welcome.
Any questions? Yeah.
So I guess we may benefit from asking questions
Maybe
later. I don't know if Kim will be able to be in the next session, but either way, there is the Discord chat for those that are attending, and maybe we can start a conversation there. I'm taking this is such an important topic to discuss, and it's really important that people are aware. I just keep thinking about gold.
Also, I was thinking about our platform in National Bank.
Okay, so with that, maybe we should just move to the next Zoom link to join all the other presenters.
I also put a Zoom link in the chat. I hope that everyone can also join this link. See you later. Thank you.
Thank you. See you later.
See you later. Thank you, Kim. Thank you.
Recording stop.